Hello guys, I hope all is well. In today's lesson, we're gonna discuss about karyotype. As an outline of this short lesson, we're gonna define a karyotype. We're gonna talk about the criteria to arrange a karyotype, the importance of karyotype, and the abnormalities that we may face while studying a karyotype. To start with, I'm gonna start with the definition. By definition, karyotype is a picture of chromosomes taken at metaphase. So, example, we have a sample of karyotype by which chromosomes we have 23 pairs of chromosomes in this photo. So the total number of chromosomes in our body is 46 chromosomes, as you already know. The first 22 pair of chromosomes, they represent our body cells, and the last pair of chromosomes, this is known as the sex cell chromosomes. This one determines whether you are male or female. Okay, so this is another example. So you notice that the chromosomes are arranged in a specific pattern by which the longest chromosomes will be the first one and the shortest chromosomes will be the last pair. So we move on in a decreasing order. Okay? So, getting back to the notes that you need to know for your governmental exams, the definition of a karyotype is a picture of chromosomes taken at metaphase. As a reminder, metaphase is a phase in mitosis by which all chromosomes are aligned at the equatorial plate or at the middle of the cell. So it is a picture, karyotype is a picture taken at metaphase. Now, after the photo is taken at metaphase, we usually arrange the chromosomes in a specific pattern. So the criteria to arrange the chromosomes depends on the size of chromosomes. That is, the longest or tallest chromosomes will be the first, and the shortest will be the last one. The second criteria to arrange chromosomes is depending on the position of the centromere. Remember that centromere is the link, or if you want, it's the link between two chromatids, the thing that holds two chromatids together. And finally, the last criteria to arrange chromosomes is depending on the banding patterns. Example, if you look into details of this chromosome, you notice that these two bands, dark in color, are aligned. This another band also aligned. These two bands are aligned. So each homologous chromosome, each pair of chromosomes should have very similar bands. They should be aligned together. Okay? So far we finished two things: the definition of karyotype and the way we arrange chromosomes to make a karyotype. Now, getting to the importance of karyotype, we have mainly two importances to do a karyotype. The first one, the karyotype is important because it tells us about the gender of the individual. That means if a female is pregnant and she wants to know whether she is pregnant with a male or female, she can simply do a karyotype. Okay? Now back to the question about determining the gender of the individual. You can simply look at the last pair. If the last pair of chromosomes is XY, then the sex of chromosomes is a male. In contrast, if the last pair of chromosomes is XX, so you have two chromosomes, that means she's a female. Okay, so don't forget that this X, it marks the position of the X chromosomes. And this is why the position of the Y chromosomes, in here we don't have Y chromosomes, so there is no Y chromosomes. This signifies that this karyotype is for a female individual, since it has XX and there is no Y. So the second importance for karyotype is it tells us whether the individual is normal or abnormal. How come? How do we determine if the individual is normal or abnormal? Well, if the karyotype has 46 chromosomes in total, that means the individual is healthy and normal. Whereas if the total number of chromosomes is more than or less than 46 chromosomes, that means the individual is unhealthy. For example, in this photo, you can notice that we have a missing chromosomes at the sex chromosome. That means we only have X at the end. We don't have XX or XY. So the total number of chromosomes is 45, and thus the karyotype of this individual is unfortunately unhealthy. Okay? Moreover, if you notice this karyotype, we notice that we have 47 chromosomes in total. How did I deduce this number directly? Because if you look at the sex chromosomes, we notice that we have XXY. That means we have additional chromosomes at the sex chromosomes. That is, the total number of chromosomes revealing in this karyotype is 47. Thus, the karyotype of this individual is abnormal. Okay, so back to the question of what is the importance of karyotype. So we have two major importances. One of them, it determines whether the gender of the individual is male or female. And secondly, it determines whether the individual is normal or abnormal. Now, coming back, or to end this lesson, we're gonna discuss the types of abnormalities that we may encounter. We generally have three different types of abnormalities. The first one is called Down syndrome or trisomy. Usually, this abnormality is done or it is placed on chromosomes number 13, 18, or 21 on karyotype. So on chromosome number 13, we will be having an additional chromosome on pair 13. That is, the total number of chromosomes will be 47. Okay? This could be in males or females. Okay? So the sex is not important in this abnormality. The second abnormality is about the Turner syndrome. Turner, also known as monosomy. That means the sex chromosomes is missing the Y chromosomes, as in this example. So, if you look at the sex chromosome, you are missing the Y in this example. This is the case of monosomy. Monosomy, that means the total number of chromosomes is 45, and thus it's the case of abnormality that is called monosomy or Turner syndrome. Okay, so in this case, you should know that in monosomy, the sex of the individual will be female. it will be sterile female. Sterile means that it can't get babies in the future. Okay? So since there is no Y chromosome, that means it signifies that the sex of the individual is female, since there is no Y. And in the end, the final case is called the Klinefelter syndrome. Klinefelter syndrome. The final case of abnormalities that we're gonna study is called Klinefelter syndrome. This is done only in males, and you can notice this abnormality when you have on the sex chromosomes there is XXY. So you have additional X and you have Y at the sex chromosome. So the total number of chromosomes will be 47. Example in this karyotype. So at the sex chromosome you have XXY. So you have additional chromosomes at the sex chromosome. And don't forget this X, this is just the place for chromosomes, the sex chromosomes. Okay? So you have 2 chromosomes at the X and 1 chromosome at Y. So you have XXY. So this is the case of Klinefelter syndrome. So this is everything that you need to know in this chapter. Please, you can ask me any questions that you want in case you have any doubts or in case you missed any idea. Thank you for listening, and be safe and take care.